Hello friends, welcome back to the sixth lecture of the microbial cell culture and its applications. You have already had studied the use of microbial cell, how to put them in the shake flask, how to put them in the reactors, how to produce products, look at the concentration of the products, then basically do the downstream processing, purify the product, scale up the reactors, look for monodes kinetics or the log based kinetics, and then essentially have a product. In today's lecture, what we want to learn and study is strain management. Now, the thing is that as you have already would have understood, or you already have an intuition suggesting that the strains or the bacterial or the microorganisms that we are going to use in the reactor plays a major role in the production of our bioproducts and then subsequent their purification. So, the success and the failure of the bioprocess to a very large extent depend on the strain ability, how much it can produce, how long it can survive, what it can do, whether it can produce in enough quantity, um, whether it can produce on continuously, what happens if there is sudden or sudden shock, like maybe temperature difference or change in the feed, a little bit change in the feed. So, that means whether the strain is robust or not. So, all of these things actually plays a major role in identifying or in the success or the failure of a bioreactor, or the product that we are want to make, or a bioproduct that we want to make. And directly or indirectly, it is also directly related to the cost. For instance, if we have a strain that can produce 100 grams per liter of titer, compared to that a strain of only 10 gram per liter of titer, that means we can produce 100 gram or 10 times more product in the previous strain compared to the later one. That means with the same bioreactor we can get 10 times more product. Hence, our overall cost of the production decreases, and that is a very major factor in deciding whether the strain is good or the bioprocess is good or not. The major challenge itself in the use of biotechnology in replacement of any of the oil based products or any of the other chemical industry based products is that whether we can produce in enough quantity at a cheaper rates, and then in that case strains plays a major role. So, that is why we want to look at the strain management today. The first part of the strain management is strain isolation. So, what does it mean? It means that whether we can select a strain that is very good among all the available strains. And when I say good, I means it can produce your bioproduct in large quantity, it is robust, it is not getting affected by slight temperature differences. And it can continue to produce, it is easy to maintain, that it does not require very expensive medium to grow. So, all of these things comes into a play. So, strain isolation is one of the factors. Second is that let us say we have isolated a strain and we found out that still it is not good enough. For example, microorganism producing 2 gram 3 gram per liter is not good enough for us, and it require expensive medium, nutrient and broth conditions and so on. So, we want to improve that strain. How do we do it? There are tools available, especially in mutation with chemicals or physical methods or genetic engineering, that can actually improve the strains. Now, let us say we have improved the strain. Now, we have a strain that is very good and we know that it works, but we also want to preserve it, we do not want to lose it, right. We do not want that it dies away. So, there are methods how to preserve the strain for years and years, so that we can keep on utilizing that strain to produce the bioproduct. Okay. So, we will have a look at all these three things. So, the first is strain isolation. If you have to guess how much amount of microorganisms are there in the world, the numbers are mind boggling. For instance, if for example, let us talk about for instance, let us talk about viruses. If you take all the viruses that are present on earth and you put them one after other, the length of that viruses, the length is more than 100 million light years. So, you can imagine how many number of viruses there are, and these are very small organisms, right. Similarly, in an ocean you have 100 million times more bacteria or microorganisms compared to stars. So, and back of the envelope calculation and estimated gas is 13 into 10 raised to power 28 times microorganisms present in the ocean. In 1 gram of soil you have approximately 10 raised to power 11 microorganisms. A human's gut, in the human gut, approximately 1 kg of is the total weight of all the microorganisms that are present in human gut. In 1 gram of dental plaque you have 10 raised to power 9 microorganisms. So, this numbers I just want to give it to you to give you a scale of amount of microorganisms that we have, and we have only characterized or did the genome sequencing of 1 raised to power or 10 raised to power minus 22% of total microorganisms that are present. So, we have not even scratched the surface. There are 99.9 times 27 or 26 times more microorganisms present. So, this is just to give you an idea that how much amount of microorganisms are present in the environment, and we have very less of them. So, the first thing comes is the sample collection. Okay. Now, you cannot collect sample from anywhere, right, means you cannot take the RO water coming in your houses or the clean water that you are drinking. You cannot take that water and basically do a sample collection from there. It makes no sense, because in that water it will be very pure, there will be no microorganism. So, what you need to do is you need to think about the conditions that you are operating in. So, for example, if you want to do a process in salt water, in sea water, which means that essentially your microorganism should be able to handle large amount of sodium salts. And why normal microorganisms cannot handle it? Because of the osmotic pressure that the salt would generate, and the microorganisms cell wall or cell membranes would get disrupted. But this microorganisms that loves to live in highly alkaline, highly salt conditions, high salt conditions, where concentration of salt is in the range of 35 to 40 grams per liter, 50 grams per liter. If you collect a sample from there, those microorganisms might be able to do the job that you want them to do in seawater. And I can give you a very quick example of what kind of job I am talking about. The job that I am talking about is, for example, flotation. It is a mining process, and you need to and it is a completely chemical process, okay, but it requires large amount of fresh water, and basically you separate the ore or the good part of the ore from the bad part of the ore or the waste of the ore. And there many times it has been, now there is a lot of research going on where they want to replace those chemicals used in the flotation process by the microorganism or the chemicals produced by the microorganism, so that it becomes relatively green mining. And in some cases the fresh water that is required is not available. So, for example, in Chile they have a lot of mines, but they do not really have a fresh water. So, there is a lot of research going on in this area where they want to use microorganisms which can actually produce and survive and grow and give products in the sea water. Similarly, in the PCR, polymerase chain reaction, the DNA polymerase has to be able to be effective at elevated temperatures of greater than 80 degrees, 90 degrees. And if you get a DNA polymerase, Taq polymerase from one of the normal microorganisms that is survives at normal room temperatures, their protein will not be their polymerase will not be able to survive out at those elevated conditions, at those elevated temperatures. So, you need to find a microorganisms that actually can survive at this thermophilic conditions, and this is what has been done by the people. They have collected the microorganisms from this elevated conditions, elevated temperature, and then basically took out its sequence and look at it, and then they use it for the PCR polymerase chain reactions. So, sample collection is very important. So, the first part would be what kind of product you want to form. Based on that, you look for production, or you collect a sample from the microorganisms, and sample means like a soil sample or a water sample or a sludge sample, or hot springs, like for example, microorganisms living in hot springs will be able to survive thermophilic conditions, or microorganisms living in surviving in volcanic areas, they would be able to handle high heat condition. There is one more example that just came to my mind. In Chernobyl nuclear reactor accident site, it is vacant now, nobody lives there. They have recently gone, or maybe not so recent, maybe in 5-10 years back, they have gone and collected some microorganism samples. They were amazed to find fungi that can actually convert or take energy from gamma radiation. And this is just to show you the capability of these microorganisms, that what can they achieve. So, if you want to use gamma radiation for production as an energy source, then those fungi are the ones where we should be working on. So, depending on, again as I have repeated earlier, depending on the kind of product that you want to produce, you need to do a sample collection. Once your sample is collected, then what you need to do is you need to put it in a nutritive medium. Okay. So, you need to know what that microorganism like. So, for instance, if that microorganism is halophilic, which likes salts, you have to put an excess of salt in that, and you have to design your medium in a way where it is happy microorganisms and happy and can grow. Okay. And basically you do it in shake cultures. Okay. Having said that, there is one major limitation here. Not all microbes can be grown in a medium and in shake cultures, that is just a fact. So, in that cases we cannot really take out those microorganisms with this kind of strain isolation techniques. Okay. So, you need to know which product you are producing, you need to know based on that product you need to select the sampling sites, you collect samples from there, then you take those samples and you incubate that samples or make them microorganisms grow in a medium which they like, and then in shake cultures. After that you enrich that the culture. So, basically what happens is that at that time maybe there are hundreds of microorganisms that are growing there, but what would happen is that as and when you start to provide the medium which is good for one microorganism that you want and not good for another microorganism that you do not want, that will lead to the enrichment of the culture. So, for example, you have microorganisms M1, you have another microorganisms M2, and you have another microorganisms M3. Okay. You want M1. Okay. So, which microorganism do you want? You want microorganisms M1. Okay. You put growth conditions GC, okay, growth conditions which are good for M1. Okay. Then let us say to begin with there were 5 of M1, 5 of M2 and 5 of M3. After this good for M1 condition, what will happen is M1 would become 50, okay, while your, so M1 would become 50, M2 will only become 10 and M3 will only become 15. Okay. So, earlier this was 33%, now it is 50 divided by 65, right. So, this is more than 80% or 75%, greater than 75%. Okay. Now, let us say we have M1, M2, M3. Okay. We want M1. Okay. Now we have 50, 10, 15. We again put them, we take some small part of it. So, the ratio remains the same, okay, of M1, M2, M3, and we put them again in a growth conditions good for M1. So, this growth conditions can be if M1 likes salt, you have salt, if M1 likes low pH, you have low pH, if M1 likes high pH, you have high pH, and so on and so forth. Now, after this what will happen is that M1 would become 500, okay. This will only become 15 and this will only become 30 or 45. Okay. Now, you see that we started initially with 5, 5, 5. This has increased 100 times, 100x. Okay. This has increased only 3x and this has increased 9x, right. So, now if you take a culture from here where you have both M1, M2 and M3, your majority of the population is M1, and that is actually what this has, what the previous one where we were increasing from 5 to 50, this one, this is enrichment, okay. And this is also enrichment, but enrichment with subculturing again. Okay. So, this is successive enrichment step, this is step 1, this is step 2, you can go after this with step 3 as well. So, then what would happen is you have M1, M2, M3, you have 500 and this becomes 50,000 because 100 times increase, right. And here it was 15. So, this will become only 45 and this was 45, this will becomes 405, 9x increase, right. So, this is what happens when you do subculturing. Now, after you have subcultured it, what happens is that we have a microorganism that is quite rich and that is what we want. Then we check it for the desired properties. So, for example, if you want it to produce antibiotics, we look at for the antibiotic properties, or we look for molecular biology techniques, that what kind of microorganism this is and whether it can produce or not. For example, if it is producing antibiotics, let us say penicillin. So, we grow this microorganism with another microorganism that does not work very well with penicillin or dies with penicillin. In that case, what you will see is that in a control where this microorganisms or our microorganism is not there, the other microorganisms M2 or let us say X2 will grow, while the in the normal where our microorganisms X1 is there or M1 is there, the X2 microorganism will not grow. So, what I mean here is if you have an M1, so this is our microorganisms, and X2 this is a microorganism to test, okay, because your antibiotic AB actually kills it. Okay. So, what does antibiotic does? It basically kills this. So, when you just grow X2 and M1 is not there, X2 will grow, right. But when M1 is there, then X2 will not grow because M1 would produce antibiotic that will actually kill it. So, that is one way of checking it. Another way of checking is the molecular biology technique, okay, or let us say we come back to the molecular biology technique, but we also can look at the product. Okay. So, another way of checking is that we have an M1, okay, it produces a product P1. Okay. Now, you have to understand that these products are they have no color or no smell, or how do you test these products. So, what you can do is you can do analytical techniques, right. You can run them in chromatography to test. Okay. So, you separate them in a column and then basically you test them, or you can do spectrophotometer. Okay. So, you can do spectrophotometer, you can do chromatography, or you can do simple titration assay or assays. Because you will have a product P, this P has to react with somewhere, right. This has to react with either with titration, means you have to, it reacts with titrands. Okay. It has to absorb light. So, that is your, it has to absorb light, that would mean that it is a spectrophotometer. Okay. Or other high end techniques would be mass spectrometry, where basically a particular mass of that product would come up in the instrument. So, this is one another way of testing it. Okay. Another way of looking at it is through molecular biology techniques, which means that you have your microorganisms M1, okay, and it has certain DNA in it, right. Okay. You know, so you can take this part of this DNA, you can sequence it and see whether it produces or not. Okay. Sorry, not produces, you can just see whether this sequence matches with your, so this sequence matches with what you want to produce. Okay. So, as you know that any protein that you want to produce would have a sequence of amino acids, and those sequence of amino acids are matched back to the ATGC or the sequence of the DNA. So, you can do a sequence of the DNA and you know that whether the sequence that you have made, does it match with the sequence, corresponding sequence of the proteins or not, and hence you can clearly say that whether this microorganisms can produce your product or not. Okay. So, that would entail the strain isolation. So, just to revise or repeat, you collect the strain based on the properties of the product or use of that strain, then you put it in a nutritive medium that basically that strain likes, and you keep on enriching it, so that the only microorganism remains is yours, and the rest of the microorganisms die or being out competed by the microorganism of our interest, which is M1. After that, you check the microorganism that it is producing through antimicrobial properties, through killing other microorganisms X2, or through product, that you have a product and you can, whatever it is producing, you can check whether this product, whatever it is producing, is the one that you want or not. And then you can also look at the molecular biology techniques like sequencing. Next part is strain improvement. Why strain improvement is important? So, as I said earlier also, that it can happen that we have a strain that is good, it is producing, but it is not good enough. Basically, it is producing, but it is not producing in sufficient quantity. Okay. Or it is producing, but it is producing intracellular, or the product quality is not good. Okay. For instance, if we are producing a protein and protein is produced intracellularly, is not folded, or there is no post translation modification done in it. So, for instance, human proteins or eukaryotic proteins are fundamentally little bit different in the way they are produced compared to prokaryotic proteins. So, proteins produced by bacteria are different than the proteins produced by humans, and if you want to produce a human protein in the bacteria, it creates certain amounts of problem, and that actually has to be seen and has to be overcome with. Okay. And most of the time the strain that you have isolated cannot really deal with that. Okay. Because those strains are natural strains, their job was not to produce that in that large quantity for commercial exploitation. Their job was to just produce it enough for themselves to basically survive very well. Okay. So, you have to do genetic engineering or you have to do mutation actually to improve the quality of the strains. Okay. Having said that, this strain improvement or genetic engineering or mutation specifically work only in the bioproduct area, where you are producing a bioproduct. But if you are using those strain for environmental engineering applications, you do not do genetic engineering. That is not approved, that is not allowed by the Environmental Protection Agency across the world, and there are reasons for that, and we will look into those reasons when we come to the microbial safety part. Okay. So, we need to improve the strain. Without improving the strain, we will not be really able to actually make a product in sufficient quantity, in a good quality, at a cost effective way. And the techniques are genetics, that we actually modify their genomes. Okay. So, the first is the mutation selection. Okay. So, as I think you are aware that whatever we want to produce is encoded into the genome of the microorganism, right. And this genome actually has to be the microorganisms would read the genome, they have this all the process of transcription, translation to produce your product or produce a protein and metabolites and all those kind of things. So, everything is encoded in the genome, and if we want to improve the strain, we actually do genome engineering. We have to change the genome to an extent. How we do it, it is a different matter, but we have to do changes in the genome actually to do a strain improvement. Okay. There is otherwise no other way actually to improve the strain in a very in a much in a very large degree. There are some other ways, metabolic flux analysis, where you can actually look at the pathways with microorganisms is producing and how you can tune it. But if you want to fundamentally change, so if you want to produce something in a microorganism that has never been produced in that microorganism, or if it is produced at a very small quantity and you want to produce in 1000 times or 10,000 times more, the only way to do is genome engineering. Now, genome engineering can be done in two ways. One is mutation selection. So, in that it is a random kind of phenomena. So, what we do is we expose the strain to chemicals. So, one of the chemical is nitrosoguanidine or UV rays, ultraviolet rays. So, this basically changes the genome of the microorganism, and then basically we take those genome, we again culture them, we see which strain or which mutated strain is a good one. So, it will not happen that the chemical or physical UV treatment that we do to a strain. So, it is not one strain, right, because it will be a lot of microorganisms, that will be a lot of same number of M1 that will be present there. And when you expose them to the chemicals, not all the M1 microorganisms would have the same mutation. So, for instance, what I mean by that is we start with M1 is equal to M1 equal to M1. Okay. So, this all three are same, okay, and they are in flask. Okay, let us say. Now, you do NTG, okay, nitroguanidine or UV. Okay. You will have M1 dash, okay, M1 double dash, okay, M1 triple dash. Okay. This M1 double dash is not equal to M1 double dash. Okay. So, M1 dash is not equal to M1 double dash, is not equal to M1 triple dash. This all three are now three different strain. Okay. Three different strain. All right. Why? Because this is a random mutagenesis. So, this chemical is used on this three at a three different any other different places in the genome, and what we get is different mutations in all this all the same strain to begin with, but now they are all three different strain. So, when you have this three different strain, so we have M1 dash, M1 double dash, M1 triple dash, and please note this M1 dash, M1 double dash, M1 triple dash is just to make them different. So, it can be M1, M2, M3, M12, 21, 32, whatever you want. Okay. So, now we put them in GC, that's growth conditions. Okay. And this growth conditions are right pH for the microorganisms, right temperature for the microorganisms, right shaking for the microorganisms, and right media component. If they like more nitrogen, then you give more nitrogen source, carbon nitrogen ratios, and so on. Okay. And then you let them grow. So, you have M1 dash, M1 double dash, M1 triple dash. Okay. Now, among all these three, one of them is going to do good, hopefully, and basically then you take this one which is doing good, and then you take it for the next steps and growth. But in many cases it can also happen that there is not only one that is doing good, but for instance this one is doing good and this one is also doing good. Okay. So, we need to proceed with that. Okay. So, now you have one M1 double dash, M1 dash, M1 double dash, and M1 triple dash. Now, you again do it with NTG or UV, okay, and it will lead to a production of M1 1 1 1. So M1 dash star, M1 double dash double star, M1 triple dash triple star. Okay. So, now all of this 6, this one, this one, this one, this one, this one, this one, these are 6 different have different genomes. Okay. So, these are 6 different organisms, or they are same organism, but different mutations in the genome. Okay. And if they survive and they grow, they can turn out to be different organisms. Okay. And this is step number 2, and it can continue as step number 3, step number 4, step number 5, and so on and so forth. Okay. So, what happens is that when you have step number 2, step number 3, step number 4, and you keep on selecting which one is good, which one is not good, and based on that basically you will get an organism of your choice, or the organism not of your choice, but organism of your choice that can make very good product, or that can have the tighter concentration as high as you want. Okay. So, one of the example is penicillin. So, penicillin production is started with Penicillium notatum. It's a and but this strain was not good, and basically after 100 mutations or so, they ended up with a strain which is Penicillium cryosogenum. Okay. And this mutation actually helped in better production, and now basically this microorganism Penicillium chrysogenum is basically used for its production. Okay. Other technique is genetic engineering. Okay. This part of genetic engineering you have learned in the previous classes also and different lectures also. So, I will not go into the details of that, but what genetic engineering does is it adds new properties and capabilities in the recombinant strain. So, for example, E. coli can be used to produce insulin, while insulin is a completely eukaryotic protein that is required for handling diabetes, but we have engineered E. coli to produce this. Okay. So, that is E. coli generally would not produce any E. coli from across the samples that you might get, they will never produce insulin. You have to add this capability in E. coli for production of insulin, for instance. Okay. So, these microorganisms can synthesize or create enhanced quantity of biomolecules. So, we can genetically engineer. So, now we just not only put a new property, but we can also put it with high copy number and strong promoter and regulatory network to actually allow it or push it to produce large amount of this molecule as much as we want. So, it is a completely synthetic pathway that we are creating there. Okay. As I said, it can lead to production of facilitate production of novel compounds. So, that means the one that is not produced in native organism ever in the smallest of quantity, we can use it to produce it. Okay. Or it is producing that, but it is using substrate that is very expensive. Okay. So, it is using very high quality, for example, glucose for instance, and we want it to use low quality of substrate. that is available, okay, malt extract or whatever, many cases just the wastewater from food processing industry, okay, which does not contain inhibitory compounds, but things like that. We can engineer the microorganism to use that, and what it does is that basically it reduces our cost. It reduces our cost to a very large extent, and this is why you do genetic engineering. As I said, we can engineer microorganisms to produce plant, animal or human protein. So, insulin, hepatitis B surface antigen. So, insulin, you know, that used for diabetes. Hepatitis B surface antigen is used for basically vaccines. Human growth hormones for helping in retardation, to avoid retardation. Interferons, we have discussed this molecule before also. So, you can engineer a microorganisms to produce these things. Okay. You can engineer not only bacteria, but also yeast. So, for example, Saccharomyces cerevisiae is a very well known organism that has been engineered to produce many things. Nowadays, Pichia pastor is another kind of yeast that is being engineered to produce. How we do it is straightforward. You use restriction enzymes to basically cut the parts of the gene that you want. Then you clone them, you have a clone them, put them in expression vectors. You basically introduce the recombinant DNA into the host cells. So, you have a plasmid. If you can do it, either integrate the whole machinery with promoter and the sequences with expression vectors and everything, either in the plasmid, or you can also include that part into the genome directly. And then you also have use strong promoters so that you can produce more. You have a regulatory switch like IPTG in lac operator, lac operon system. So, all these things are there, and we basically use this to improve the strain. There are also challenges in the strain improvement. I think you already have known that, right? The challenges are that it is not straightforward. It takes a little bit of more challenging part. So, for example, if you want to clone a eukaryotic protein into the prokaryotic one, then also you need to excise non-coding regions. Okay. That means that it needs to be the mRNA needs to be changed to cDNA, and then you need to do an excision, and all those kind of things are necessary. Another part that I have already talked about is that the human proteins or the eukaryotic proteins undergo post translation modification. Okay. It is just a process where they modify the protein that is produced through the cellular machinery itself, but that does not happen in the prokaryotic cells. Okay. That lead to inaccurate foldings, that leads to formation of inclusion bodies. These bodies are inside the cells, not released outside. So, we have discussed that part, that if it is inside the cells, that means that you need to do a cell rupture, and your downstream processing becomes extremely expensive. So, you also need to essentially to take care of these things when you do a genetic engineering. Okay. In the case of, and this is particularly for the case of production of eukaryotic human protein in bacteria. So, you can produce human protein in Saccharomyces cerevisiae or Pichia pastoris to overcome these challenges. Saccharomyces cerevisiae is a very good organism. We know it very well. It has been used to produce many of the human proteins, and we will see this in the coming slides. In this lecture or the next one, it has been used to do that, but still it lacks, that it does not produces more than 5% of the protein, or the concentrations are not good. So, that is why people have started to use Pichia pastoris. It is also a yeast. It is used to produce this human protein. It does very well. One good thing about it is that it does not produces extracellular proteins, its own extracellular protein. So, it is actually in a way very good to use this. So, there are tricks available for strain improvements. You use mutation to improve it, you use genetic engineering to improve it. You can produce and add new capabilities like production of insulin or hepatitis B, antigen in E. coli, and means you can do that, but it also has challenges. So, non coding region is one of the challenge, inclusion body or inaccurate folding is another challenges. You can use eukaryotic organisms like Pichia or Saccharomyces cerevisiae. People also are planning to use mammalian cell cultures. This is another techniques that is coming on, but mammalian cell culture require extremely expensive medium, and that is also another challenge. So, we want to do it in a in microorganisms that are cheaper to operate and run, and that's the way we would like to that basically at the end of the day the idea is to decrease the cost, and all these things are done to decrease the costs. Okay. So, for instance, as I was saying, mammalian culture would have been straightforward, but then running a mammalian culture is extremely expensive. Okay. Then another technique for strain improvement is basically metagenomics. Okay. So, metagenomics is a technique where you take the sample from the environmental samples and you isolate the DNA. So, you isolate a bulk DNA, you do not segregate the microorganism and you isolate that bulk DNA. After that, basically you do the restriction enzymes or the cut the DNA, excise the DNA at you with some restriction enzymes that you already know, and then basically you ligate all this DNA into the expression vectors. So, this is step number 3. Okay. After that, you have different plasmids that are available, and basically you construct a library, you put those plasmids into the microorganisms in E. coli for example, and then you see the expression of proteins from each of this different E. colis and analyze, and then see basically how good is the expression and how good is the expression and how good is the analysis. So, so basically you have maybe just to make it a little bit more clear, you take the DNA from the samples, okay. You do extraction. So, you do not get the DNA, you get the microorganisms. Okay. So, you get the microorganisms from the samples, environmental samples, you extract the DNA. Once you have extracted the DNA, you basically do restriction enzymes RE, okay, or maybe I can write it full. Okay. You do the restriction enzyme. So, that means your DNA is now fragmented. What you have is a fragmented DNA. It's okay. Once you have this fragmented DNA, basically you ligate, so join them. Okay. You are aware of these terms from your previous studies. So, you ligate this restricted fragmented DNA after restriction enzyme, okay, and in basically in another expression vector. Okay. Now, this expression vector is basically transformed or put it in E. coli. Okay. Now, once you have, so now you have an E. coli cell. Okay, this is E. coli with your fragment. So, this is fragment 1, you have another E. coli and this is with fragment 2. Okay. This is with fragment 2. You would also have another E. coli. So, you have multiples of E. coli, right. You have many of those E. coli, but and this is with fragment 3. Okay. Now, basically use them, put them into the growth condition. So, good conditions that you want, and you check the expression. Okay. And you check the protein production or your product production, the normal methods of checking. Okay. So, this is another technique that is available in the metagenomics. So, in this technique there are several advantages. Uh, the advantages are, first it is a high throughput technique. Okay. So, you can actually do with a lot of microorganisms in one go. It does not require that we wait for, it does not need, it does not require that there are takes, means you have to do individually one by one. So, it is a high throughput technique. Second is that it can cater for both culturable and non-culturable microorganisms. As I have explained earlier, that we need both, many times from the environmental samples you will not be able to culture those microorganisms in the shake flask or in the petri dish plates. However, in this technique, since we take only the and only the DNA part and then grow it in put them in the E. coli and grow the E. coli and then analyze, then we actually overcome this limitation of culturable and non-culturable microorganisms. As I have explained, the steps include restriction, so DNA extraction, restriction, digestion, cloning, and then expression. And then after expression, product evaluation or protein evaluation, concentration measurements using either titration, spectrophotometry, HPLC with chromatography, or MS. Okay. Now comes the last part in the strain management, that is strain preservation. Okay. So, now you have a strain that you have identified that works very well, and you have done all the hard work to analyze that strain, characterize that strain, and you know that it is very good and works very well, but you also need to preserve that strain. Without preservation, in let us say 1 year from now, 2 years from now, you are running a reactor, let us say your reactor something happened, let us say the pH management system or the pH management controller on the reactor fails, and the reactor become acidic, and instead of operating the reactor at pH 7, the reactor is operating now at pH 2, and all the microorganisms and every product and every batch is completely destroyed. Okay. Now, you have to restart your reactor again, and you do not have your strain because your reactor is dead. Then it becomes very tricky. If you have not done it, you lose all the work that you have done so far. So, it becomes imperative that you do a strain preservation. So, you have a people not only do it one strain preservation, but they have multiple strain preservation at multiple locations, and this is really important and really needed to be done if you want to save your work. If you want your reactors to perform continuously, and in an industry this is extremely important. So, having said that, what are the ways of strain preservation? One is storage on agar. So, here basically you grow the microorganism on the agar slants and or stabs, and basically stored between 5 to minus 20 degrees. Okay. This kind of storage can stay well for approximately 6 months interval, but after 6 months you should be actually keep on growing them again and checking that if they are okay, do the sequencing, find their DNA, check their product and everything, and then basically again do the slants or the stabs and then again save it. So, this has to be done on a continuous basis. Another way is basically you actually grow the microorganism. So, you normally grow the microorganism, then add cryoprotective agents like glycerol, okay, 10 to 30%, many times people can increase this to 80% glycerol stocks, and then basically you put them in sealed ampoules and freeze them in liquid nitrogen. Okay. So, the temperature is somewhere between minus 176 degrees to minus 196 degree. In our labs, we actually keep them at minus 80 degrees glycerol stock with concentration of around 50 to 80% of glycerol. We take 1 ml or 200 microliter of our culture and 80% glycerol, or this percentage can vary. It is not very necessary to be the percentage to be accurate, and we store them at minus 80 degrees, and that also works very well. Another technique is lyophilization. This is freezing of a culture by drying under vacuum. Okay. So, as you know that the pressure and the drying point or the boiling point are dependent. So, as you basically decrease the pressure, the boiling point or the boiling point of the water also decreases. So, here basically what is being done is you remove the water from the culture by essentially working it at a low temperatures as well as under high vacuum. So, you really decrease the pressure, so that you allows the water to get out at a very even at a room temperature. So, even lower temperatures, even at 4 degrees you can get rid of the water under vacuum, and that is what process of lyophilization is. The way it is prepared is then there is no water in it, okay? And since there is no water in that, basically the microorganisms or the culture actually survives and remain viable for 5 to 10 years. And the good thing is that you can actually store them at room temperatures. So, if you have lyophilized culture, you can store it at room temperature in a sealed packs, and there would be no problem there. So, this is one of the way how strain is preserved in the preservation this culture banks. Okay. So, for your lab you are preserving, right. So, whatever you are working on or your product, you are preserving your strain, but many times it happens that you want to work for let us say you have lot of strains and you are working with only 3 strains at the moment, but rest of the strain you are not working. But if you have to preserve them and if you have to revive them every 6 months and check and everything, it is a lot of work. So, what has been done is that there are lot of strain preservation culture collection centers. Okay. So, these are the culture collection centers which are there. One is ATCC, American Type Culture Collection, which is in United States. There is NCIB, National Collection of Industrial Bacteria in UK. So, you can order them for research purposes. There is no actually problem to order from them. There is a DSM in Germany. In India, we have MTCC in Chandigarh. This is Microbial Type Culture Collection and Gene Bank. And we have NBAIM, National Bureau of Agriculture, agriculturally important microorganisms. So, these are there, and if you want them, you can just pay a very small amount of fees and you can order them, and the culture would come in a lyophilized form. You can revive them, and there are a couple of others also. They can actually give you in what form you need, and you can basically use them. So, in this lecture what we have seen so far is that we have looked at the strain important. So, first part is that why strain is important. This is obviously have been clear to you now that without a good strain we cannot really move forward. So, we need really a good strain. Now, to get a good strain, one option is of course that we actually take it from environmental samples, and this we can do it if we know what kind of product we are looking at. So, this has been done through either halophilic condition, hot springs, volcanic conditions, and different other conditions that are available. So, you take that strain. Now, you give them a good conditions and enrich them so that the strain that you want is actually is getting enriched and it is getting more dominantly present. You check whether your strain that you want is getting more dominant or not by essentially taking the strain and seeing what kind of product it is producing. Okay. If it is antibiotics, you can put it with another microorganism to say that whether this antibiotic production is enough to kill the other microorganism. So, kind of checks. You can also do molecular screening, DNA sequencing, and so on. So, that is strain collection. Okay. Then the next part was strain improvement. For strain improvement, you can do mutation, you can do genetic engineering. There are challenges with genetic engineering, but of expression of proteins in human proteins in microorganisms, and that is not that easily handled, but there are tricks available. You can do it in eukaryotic organisms like yeast, Saccharomyces cerevisiae or Pichia pastores, and you can do genetic engineering there as well. So, there are tricks available how to overcome the inclusion body challenge, how to overcome the known coding sequence in the sequence of your interest. So, those are the things, and then comes the strain preservation. You need to preserve that strain because you do not want to lose it, and for that essentially you need to either do it in agar plates, or you do glycerol stocks, or you lyophilize it, and you make sure that they are available to you if you need, and then basically there are also collection center that you can donate to them. They will maintain it for you, and then they are available as and when anybody requires. One important point in this strain preservation is for research purposes it's easy to get, but in case you have to do it for commercial purposes, you have to take care of the IP issues. So, strain itself, if the patent can be filed on that strain, and even when it is at strain collection centers, culture collection centers, that IP is protected, and IP is intellectual property. So, the intellectual property is of the individual or the institute where that strain has been made, and they have filed a patent for it, and with that patent it is protected. So, for research purposes it is not really that big a problem. You can just sign material transfer agreements and you can get the strains, but in case you need to use it for any commercial related activities, you need to get over you need to deal with the IP issues, and you need to have a licensing agreements or this kind of agreements. So, with that we will end this part here, and we will have another lecture, the last lecture for this unit, for good microbial practices, the applications of where this the cell culture techniques have been used, and what are the do's and the don'ts for the microbial culture practices and so on. So, thank you very much. We will meet again for this lecture soon.